Morning. The lecture that we will discuss today is about biology. Of course, this lecture is a lecture about biology, but it's for physicists. So I will try to keep it as simple as possible and as relevant as possible to the radiation biology that we will start from the next lecture. However, I realize that some concepts are a bit complicated, but I want at least to give you a flavor, a hint of what is the current status of the biology. It's not, let's say, I don't want to simply discuss, the 20th century biology. I want to give you a glimpse of the 21st century biology. So it also, I have to say, it's a very long lecture, of course, which means that in the video I split it in several pieces, and also when you study it, I recommend it to go slow, not to see the whole 100 slides that you will find in the Moodle webpage. So, in fact, I want to start with radiation biology. Let's say why we need to know biology. We need to know biology because the target of radiation action, the reason why we have radiation effects when humans or animals or cells are irradiated is principally the DNA damage, because DNA is the largest target in the cell, is present in 2 copies only, and is essential for cell regulation. So all activities, not only what is written in the DNA determines how we are built, but is active all the time, because every time there is a need for a reaction to an external stress, you need to read the DNA. Membrane damage, of course, you can say that the membrane, is the envelope that includes the cells. In principle is also important because, of course, damage to cell membranes disturb the state of cells' electrolytes like calcium, and if it is not increased, you can induce apoptosis, which is a form of cell death. We will see this when we will see cell killing. But generally, membrane damage is important only at very high doses. So when the dose is so high that you can actually really damage large fraction of the membrane, and you induce structural damages in the membrane. And the cytoplasm, which is the medium between the membrane and the nucleus where the DNA is of course essential for the metabolism of the cell, but again, damaging different components of the cytoplasm, they can be replaced by the DNA itself. However, the damage to mitochondrial DNA, so we have mitochondria, and in the mitochondria there is a DNA, this can cause late effects and oxidative stress. Mitochondria are the most important engines for the energy of the cell. So this means that we will essentially look at all of these fractions, but at the beginning we will start more with the molecular biology at the subcellular level. So we will see the different components of a cell, and then we will assemble them together to see how the cell behave all together. So let's start with the membranes. The membranes, this is something that in physical chemistry is very easy to do, because you can actually build the membrane that has nothing to do with biology. Let's say if you take phospholipids are these molecules that have a head which is hydrophilic, and then they have a tail which is hydrophobic. It's actually 2 tails, you see. So hydrophobic is like an oil, and hydrophilic instead means, of course, that is lightly charged. I mean, it's not ionized, but there is an excess of charge. And because, as you know, the water molecule is also not neutral, it's charged, because there is more electronegativity around the oxygen than around the hydrogen tails. Of course, every molecule that has an excessive charge will nicely integrate in the water, while every molecule which is completely neutral with no charge will be expelled because the water tends to stay with itself. So spontaneously, if you put phospholipids in water, they will take this configuration. This is called the hydrophobic force. It's not really, of course, it's always electromagnetic force, but it's called hydrophobic because it means that the molecules that are not making any kind of binding with the water will be excluded by the water, and eventually they will assume a configuration like this, where the internal part has no water and the external part, the heads, is interacting with water. And this is, of course, spontaneously. Only the hydrophobic force is responsible for life, if you want, because, of course, only because of hydrophobic force you can make these nice membranes. And of course, only if you have a membrane you can have life. Let's make it straight, because as you know, life goes against the entropy law. So in life, you want to make more order against the rest of the world that goes in more disorder. And to do that, you have to separate the environment. You must work in a separate environment, which is reducing the entropy, of course, according to the second principle of thermodynamics, at expenses of the external world, which is still increasing the entropy more the time decreasing the entropy in the internal part of the cell. The actual structure you see of the membrane, as you see here, this is the total mammalian cell here, and this is the piece that we are talking about now, the membrane. Is actually much more complicated than a micelle. This is also called micelle, a phospholipid double layer that you can build just in the lab, just using phospholipids in water. In fact, you see, this is the double layer. The double layer is between 3 and 5 nanometer thick, so it's pretty thin, actually. And you see that there are a lot of molecules that actually are, some of them on the surface, like in the external surface, some of them in the internal surface, and some of them are transmembrane, so it goes through the membrane. They are absolutely essential, because these proteins are the only way that the cell has to exchange material with the external world, and also to communicate with the external world. In addition, there are many other molecules. A lot of oligosaccharides, for example, are attached to these proteins, and many other stuff. So imagine this layer, this envelope, but imagine also many other proteins. We will see what are the proteins in a few slides, but this is just for starting. From the physics point of view, there is a large part of biophysics, actually, which is really dedicated to the membranes, because there is a lot of physics in the membranes. And when you study the nervous system, the central nervous system, but and the peripheric nervous system, the physics, especially electromagnetism, is completely dominant in the study. So physiology is really based on the Maxwell equations, if you want. Why is that? This is because actually, as you see simplified here, let's say the cell membrane is really a capacitor. Is really a capacitor with an external part which is positive, and internal part which is negative, and the potential difference between the voltage, let's say, of the membrane in mammalian cells is around 70 millivolts. It can change very much. Why is that? Why it happens like that? Well, the reason is quite simple. I mean, all the cells have a system which is called the sodium-potassium pump, which is actively pumping potassium inside the cell and expelling sodium from the cell. So essentially, the external medium, you see the water here, is rich in potassium, and of course it's rich in chloride. It's rich in sodium and in chloride. Sodium chloride is salt. So all water, you know, is rich in salt. So in the external medium, you have a lot of sodium chloride. In the internal medium, you have a lot of potassium, because the sodium-potassium pump is actually generating a disturbance in the concentration. So it's accumulating potassium inside. Now, this potassium and sodium, they are so small that they can go through the membrane. So this means essentially that from the internal part of the cell, from here, the potassium will have a tendency to go out simply because there is a concentration gradient. So if you write the diffusion equation, you will see that all the potassium has a tendency to move for the concentration gradient from the internal part to the external part. But, and this is of course interesting, the moment that they tend to go out, they leave a negative charge inside the cell. Because if there is a charge equilibrium in the cell, the moment that the potassium tends to diffuse, you leave a negative charge on the other side. And then you have an equilibrium when the force, the electric force now, balance the diffusion force, let's say the concentration gradient. So the flux due to the concentration gradient is balanced by the flux due to the Coulomb force. And if you balance the two, you come out with the famous Goldman equation here. Of course, in the Goldman equation, I only made the case for potassium, but you have to consider also sodium and chloride that instead have a tendency, of course, to go in. But then, if you look at the final equilibrium state, let's say where the electric force balance the diffusion forces, you come out. I'm not going into detail, but this is relatively easy. It's done in non-equilibrium thermodynamics. Is a typical exercise. I think you will do it in some statistical mechanics course. Then the potential difference is given by this equation. You see where, of course, R is the gas constant, T is the temperature, F is the Faraday constant, and P is the permeability. Is the permeability of the membrane to the different ions. Of course, if a permeability is 0, there can be no diffusion. So, of course, the permeability is an important parameter. And these are the concentration, external and internal, of the different ions. Now, if you solve this equation with typical values of the concentration, you will come out with the value around 70 millivolts. This means that if you calculate the electric field, which is of course the potential divided by the size of the membrane, which is only a few nanometers. So here we use a value of about 7 nanometers. You come out with a very high value of the electric field. It's 10 to the 7 volt per meter. So it can be a low potential, but it's very high force. And the capacity of the capacitor at this point can be easily calculated. Here, this is the usual formula. This is about 50 picofarads, which means approximately 1 microfarad per square centimeter. The change in this potential is what is responsible for the functional activity of the neurons, is what is responsible for the contraction of the muscles. This is all completely based on the change of potential in the membrane. So it's, of course, it's a huge topic in physiology. We will not go into these details here. We will focus more on the DNA, but membrane is also important. So at least I want you to know that this is how to deal with membranes. Of course, as I said, the most important part is the nucleus, and the nucleus is full of DNA. The total length of the DNA is in the order of 1 meter, is actually more close to 2 meters, and the size of the nucleus in mammalian cells is 10 microns. So you have 2 meters packed in 10 microns. So you can imagine how much wrapping you have to do to pack this very long string in only 10 micron sphere of approximately 10 micron diameter. It's very long string, but it's very thin. It's very thin. The thickness you see is in the order of 2 nanometers. Is in the order of 2 nanometers. And the number of the structure is also relatively simple. there is a sugar phosphate backbone. So here there is, you see, sugar phosphate, there is a sugar and there is a phosphate. This make the external structure of the DNA, the so-called strands. Between the two strands are kept together by the bases. There are 4 bases in the DNA, guanine, cytosine, adenine, and thymine. And the forces that keep the 2 bases together are not strong forces, you know, are not ionic bonds or covalent bonds, are only van der Waals forces. So it's a relatively weak force here that keeps together these base pairs. You already know, of course, that the sequence of the bases, guanine, cytosine, adenine, thymine, is the genetic code. We will see this in a while, but I'm sure you know it already. Keep in mind that the number of base pairs in a human cells is 3 × 10⁹. So we have 3 giga base pairs in a human cells. So this is like the number of characters in a book, because we see reading these letters, let's say, is how we know the structure of an organism. So we are talking about 3 billion words written in our DNA. 3 billion characters, to be precise, not the word, you know, because as in the language, to make a word, you need many characters, and that's the same for the DNA. 1 single base pairs is not enough. You need a sequence of base pairs to make a word, to make something that then has a meaning, and we will see in a second what is the meaning. So this is a little bit more detail on the structure of the DNA. This 1, 2, 3, 4, 5 is the position of the carbon atoms in the sugar. So you see, this is oxygen, is shown in this light blue. In green is the phosphorus, which is essential because you see the phosphorus keeps together the different sugars. So it's the link between the sugars. And the carbons are shown here, 1, 2, 3, 4, 5. So you see, it's very interesting. So this is the sugar phosphate backbone on one side. So if you go back to this figure, you have 2 strands. You know, the DNA is typically double-stranded. So on the one strand, you have this kind of configuration. And on the other strand, these are the bases that keep together the DNA with these hydrogen bonds. So it's a relatively weak bond here, but keeps together the DNA. On the other strand, you see it's exactly the same, but the other way around. This is extremely important. So this is called 5 prime, 3 prime. You see, from the carbon 5 to the carbon 3, this direction, and this is called instead 3 prime, 5 prime. So if you go here, you go from the carbon 3 to the carbon 5. So one is the mirror image of the others. It's like having 2 hands in this way. And there is no other way you can do the DNA. So if you do the DNA, you have to do it respecting the 3 prime, 5 prime, 5 prime, 3 prime law. So this is very important also for the recombination. If you have a damage in the DNA, how you have to rebuild the DNA. You must always respect the 3 prime, 5 prime, 5 prime, 3 prime law. It's like having, you know, 2 strands that are not like that, but are like that. This is a movie that is actually a software that you can use to see the DNA in 3D. It's you know, you see, it's nothing special these days. This is not very new, but you see, you can actually zoom, and now you see from the strands, you see the nature, for example, of the bond. That's interesting. You also see the nature of the bases here and the hydrogen bonds, and you also understand why there is only. Yeah, this is now you see the orbitals of the bases. you also understand why there is a law, there is a very precise law in how the bases can couple. It's not that every base can couple with any other base.
The chemical bonds. Finally, I can see the chemical bonds.
You see here the chemical bonds. So the adenine can only be coupled with thymidine, and the guanine can only be coupled with cytosine. So you have GCAT bonds. It's not possible to have anything else. It's like a key lock mechanism. You see, the structure of the basis is such that you can only build it having AT and GC. Okay, you can watch the movie. I uploaded, in this case, you have noted, I uploaded the full Microsoft PowerPoint file, so you don't need to watch the movies offline. You can, they are, let's say, inside this presentation. So now this is probably the most important slide of the lecture. And it's called the central dogma of molecular biology. This is due to Watson and Crick's, but it is about in the 50s, between the 50s and the 60s. So it's now many years ago, about like 70 years ago. It's called dogma, which is a word that in science is never used. Is used on purpose, let's say, it's almost ironic if you want. And in fact, we will see, it's not completely true, as all dogmas are not completely true. However, I think it's nice to call it dogma because you have it in mind to say, okay, this is how it has to be. So every time you find exceptions, you are correctly surprised to say, oh, there is something strange here. So what is the dogma, the so-called central dogma of molecular biology? The central dogma of molecular biology is that the living organisms work in this way. They work from DNA to protein. So this is the book, and these are actually the actors. These are the molecules that do the work, that make us living. Everything is written in the book. And the arrow goes strictly in this direction, from DNA to protein. This arrow here means the DNA is able to self-replicate. So the DNA can make a copy, the replication, can make a copy of itself to pass it to the daughter cell. So this allows the growth of the cells and eventually of the organism. So the dogma is that all the information is in the DNA. The DNA contains all the information about the structure of the cells, the structure of the tissue, the structure of the organism. Is everything written in the DNA according to the code that we will see in the next slide. I told you already, is the sequence of the bases, essentially. The second step is that the information must be read, and the reader of the book, the molecule that is actually reading the book and decide, okay, this is what is written here, is the RNA. The RNA is a molecule very similar to DNA, very similar, same structure, but two main differences. One is that it's single-stranded, it's not double-stranded. And the other one is that there is one base is different. There is no thymidine, there is uracil, but this is a slight difference because uracil has the same structure of thymidine, so it makes still the same kind of bonds as thymidine. So what happens? It happens that you have to open the DNA to read. It's like a book. To read a book, you have to open. So to read the DNA, you have to open, but this is not so difficult because, as I said, the hydrogen bonds that keep together the strands are very weak. At this point, the reader is built, and the reader is the messenger. It's called messenger RNA. We will see that there are also other RNAs. Now, this is built with the bases complementary to the bases that I'm reading here. So every time there is an adenine, I will put a uracil. Every time there is a guanine, I will put a cytosine, and so on and so forth. At this point, I make a string of a given length, because of course it's not the whole 3 billion base pairs that are read. It's only few thousands, generally, if not less, of base pairs that are read every time. This strand now contains the information, and now comes the next step in dogma. It goes out from the nucleus, goes into the cytoplasm. So we leave the DNA alone, and we are now in the cytoplasm. And now there is someone that needs to translate. So this is called transcription. Now we need some translation. So the reader now has some information. It's like having the plan to build a palace, to build a chair, to build a table. But now someone has to take this take this information and actually has to build this chair based on the information that the messenger, is called messenger not by chance, because it is transporting the message from the DNA to the cytoplasm, is bringing. Who is reading the reader now? Who is translating? The main character here is the ribosome. The ribosome in the cytoplasm is actually scrolling through the messenger RNA and translates the information into proteins. So going through the messenger RNA, it generates a protein, and then the protein is the one that will be a structural protein to be part of the cell, or an enzyme, which is facilitating chemical reactions. All kinds of things are done by proteins. So essentially, the proteins are the product of the DNA message. As I say, this is the central dogma. So you always have to think DNA, RNA, protein. You think that this is true very, very often. We will see that, as usual, the things can be much more complicated. So, of course, this is what we call now classical genetics, if you want. In modern genetics, we will see that the regulation of the information, so the information in DNA can be manipulated by the proteins themselves, and this is called epigenetics. So in addition of this, there is epigenetics. But I think this is clear, important, and you have to remember this as a basis. And this is probably the message that I want you to remember. That the central dogma says the information is in DNA, the transcription, so the reading, is done by the RNA, the translation, so the construction of the real building blocks of life, which means proteins, is done in the cytoplasm by the ribosomes that are reading the messenger RNA. So we said the ribosome translate, no? They translate the code, but how do you read the code? So the proteins are a sequence of amino acids. So there are 20 amino acids. There are this molecule like tryptophan, phenylalanine, glycine, serine. These are these molecules that, put in a sequence, make a protein. There are 20 of them. There are 20 different amino acids, and there are 4 bases. So how many bases you need to define an amino acid? Of course, if one base was corresponding to one amino acid, you will have only 4. There's nothing to do. You will have only 4. If 2 bases corresponds to a given amino acid, how many you can have in that case? Well, in that case, you will have you have 4 bases, 2 of them, so 4 to the 2, this is 16. So it's almost good, but not good enough. So you need 3. And in fact, you generally use 3. Now, the combinations of 3 bases having 4, which is adenine, uracil, because of course you are in the RNA, guanine and cytosine, make 64 combination, which is more than 20, which means that the biological expression is to say that the code is degenerate, is redundant. So this means that several triplets give the same amino acids. So you see, the way to read the book, the Rosetta Stone, I mean, the translation of the DNA reading into proteins is here. You see, for example, if you have a triplet of CAU, cytosine, adenosine, uracil, you will have histidine. Of course, also CAC, cytidine, adenine, cytidine, will give again histidine. But for example, cytosine, adenine, adenine will give you another amino acid, and so on and so forth. So this is what the ribosomes do. The ribosomes translate the code into amino acids, which means proteins. Of course, there must be a beginning and a start. You realize that you don't read the whole code, you only read the piece. And somehow, you have to know where to start and where to end. This is what we call the gene. The gene is a very old concept, is much older than the concept of DNA, of molecules, of strands, of genetic code. Before this, people already realized that there are genes, there are units that we can transmit to our offsprings. And now we know that this is the concept of gene. Every gene will be a sequence of DNA that transcribe for a given protein. Now, and as I told you, there must be a beginning and there must be an end. And this is what is interesting here, because you see, how do I know where to start and how do I know where to stop? This is actually how the structure of the DNA really looks like. The gene is essentially here. You see, starts from here and ends here. Okay, this is the gene. So this sequence here is the sequence that code for one given protein. Before this gene, there is a number of sequences that are not coding for the protein, but they are generally called enhancers. So these are regions that tells to the enzymes this operation, let's say, of building the RNA is done by proteins, actually. These proteins are called enzymes, and in this case are called RNA polymerase. Polymerase means that I build a polymer. So the RNA polymerase is building the RNA polymer. How does he know where to start? He knows because he starts to see this enhancer sequence, and then he starts to see a promoter sequence, and then he starts to transcribe. Now you see, even here, the situation, and then he knows where to stop. This is the stopping sequence. The stopping sequence is a poly-A in general, A. Before there is a stop codon and the trailer. But the interesting part, you see, is that even in the gene, there are some regions called exons and some regions called introns. The introns are not coding for the protein. So there are two RNA, actually. You see that already the dogma is wrong with the new knowledge that we have now. There are two RNA. One is the pre-mRNA, the primary transcript, that as a start, as an end, but includes these introns. Then there is a further processing called splicing, where the introns are removed, the exons are put together, and now you have a complete messenger RNA. And the messenger RNA, again, contains a starting cap, a leader, a starting codon. Codon is a triplet. So I can say codon or triplet in the same way. So this is the starting codon. Then there is a stop codon, a trailer, and a poly. This will be now the structure of the gene that is read by the ribosome. The 5' cap is read always in 5', 3' direction. This is also extremely important. The 5' cap and the poly-A are more is technical issues. So they are molecules that are used by the ribosome to capture the RNA here and to release the RNA here. And then, but the gene itself is only this piece down here. So you see, this is explained in great detail. Now I will not read it because it's, as you can imagine, is extremely complicated. It's and is also a knowledge which is often evolving, you know. But you see, something that for example you should know is that in eukaryotes, which is our cells, in mammalian cells, the promoter is at approximately -25 base position, and the consensus sequence is TA, 5 prime, 3 prime. This is called Tata box. So these are the sequences, let's say, that the RNA polymerase recognize as promoters. There are also, you see, other sequences and other probably you can read this one. Of course, how the RNA polymerase recognize the sequences to translate the right gene is a very complex model. And if you read this slide, I mean, you will find more details in it. And this is now a little movie that shows you how essentially
Here is a cell, the basic unit of all living tissue.
We can watch it together.
In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes.
This we will see later, okay? So don't
Each
worry.
chromosome contains a long strand of DNA.
The important thing is that you know that there is a long strand of DNA.
Called histones.
And there is also proteins that bind the DNA. But this is something
within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA.
So you see, this is the real start of the transcription, when you have this RNA polymerase, which is now reading the DNA.
A strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order.
Of course, I forgot to tell you that to build the strand of RNA, he has to have bases, free bases, which are not the bases on the DNA, but other bases that the RNA polymerase can bring along and can somehow attach to the different to the different bases on the DNA, always following the Watson and Crick rule of complementarity. So you see, every time you see a C, he puts a G, GC, TA, CG, and so. And here is uracil, for example, because there is an adenine, you should put a thymidine, but then it's replaced by uracil.
In which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
This is what I told you, the splicing. So you have to remove the introns and reattach the exons.
The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome.
So you see, now there is another type of RNA. This is called tRNA, that you mentioned before. But to help the ribosome in doing its job, you need someone who is able to read the code, of course. And now, in a way, you reread the code, which is still complementary to the RNA code, which means that now the code is identical to the DNA code. And for every triplet, for every codon, the tRNA will bring the right amino acid.
The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so I think you get the point. Now, is all DNA coding? Is all DNA making genes? We have seen that this is not true, right? We have already seen that it is not the case. We have seen that there are pieces that are enhancer, there are introns, and so on and so forth. However, it's interesting that in humans, there are 30,000 genes in total, and they code for more than 100,000 proteins. So the one-to-one correspondence gene-protein is clearly not true, is not possible. You have less genes and proteins. Moreover, if you take the difference in the DNA between human and an ape, you know, like a chimpanzee or an orangutan, the difference is less than 2%. It's 1.8%, something like that. And so far, all the genes have been found in both. So 30,000 genes in humans, 30,000 genes in the ape, difference of only a few percent, but one is a human and one is an ape, which is, except for some of my friends, is a quite evident difference. Now, 30,000 genes is a very low number, very low. Is only 2 times the number of genes in Drosophila, the fruit fly Drosophila, the common fruit fly that you always see on the fruit, is one of the most important genetic organisms in classical genetics. And it has approximately 15,000 genes. So it's not this huge difference, but we feel very different from the fruit fly. Besides, given the length of the human DNA, this means that about 98%, 3 per 10 to the 9 genes, is 3 per 10 to the 9 base pairs. Now, if you take the average length of a gene, you find out that approximately 98% of the DNA is not coding. And this is, let's say, junk DNA. So you have, and this is also interesting, you see, the junk DNA is bigger when the life becomes more advanced, more evolved. If you take prokaryotes, we see prokaryotes are bacteria, single-cell eukaryotes, plants, these are octopus, chordates, mammalians, and then humans. You see that the fraction of junk goes up, up, and up. Humans are the one with most junk. So what is the role of this junk? Probably there are two answers. I mean, one is simply evolution. So one is simply areas of the DNA that were initially were coding, and then they were not coding anymore, but they are still there. Another one we will see is the regulation of the genes themselves. We will see also with the introns. The introns we have seen before are apparently junk, because they divide pieces of the gene, but we will see that they are not necessarily useless junk. So if you look then at the components of the human genome, as you say, there is about 2%, only 2% are protein coding genes. So the 30,000 genes are essentially here. Then you have a large number of other stuff. And you see, the other stuff include introns that we have seen already. So there are more introns than exons. You see, so there is a lot of material in the genes, not outside the genes, but in the genes that are that is dividing this in the genes. Then you have the so-called the interspersed nuclear elements, the lines, and the short interspersed nuclear elements, the senes here. This is a lot of stuff, you know. So these are lines of the DNA. Some of them are very long, the lines. Some of them are very short, are less than 500 base pairs that normally are not genes, but in some cases, especially the lines, can be transcribed to make reverse transcriptase, a type of enzyme, or endonucleases. Then there are long terminal repeat, the so-called LTR, that are here. There are retrotransposons that can amplify themselves through an RNA intermediate. So these are pieces of the DNA that amplify themselves. And the so-called jumping sequences. These are sequences that can change their position in the genome itself. These jumping genes are important in the DNA fingerprint, for example, because they are always located, they are always different in different individuals. Generally speaking, these interspersed DNA are exactly those that are sequenced in the DNA fingerprinting, because those are the areas with maximum variability. They don't code for genes. The genes instead themselves, they are very, very similar between the different individuals, because at the end we are all humans. Not the same, but we are all humans. Now, as I told you, the introns that make up some almost 30% of the human genome, they seem to be useless, but in reality, they can be instead extremely important, because you can have what is called alternative splicing. So you see, through a large number of mechanisms, you can have exon skipping or cassette exon, mutually exclusive exons, alternative donor sites, alternative acceptor sites, and intron retention. You can have different splicing. So the same gene with the same exons and the same introns can then generate, using alternative ways of rearranging the exon, or in some cases, even keeping an intron in the genes in some cases, you will end up with different protein isoforms or different proteins at all. And this partly explains how you can do 100,000 proteins out of 30,000 genes, because one gene can be spliced differently, and then one gene can give up to different proteins. But the important thing to understand here, which is really essential for our everyday life, is how the transcription is regulated. So how can this be? I mean, of course, you can imagine the transcription cannot be always the same. You can imagine that we all come from a single cell. This single cell was a fusion of a sperm cell and an egg cell. Okay, was one. Now we have billions of cells. What happened? So at the beginning, you had to use so many genes to do so many things that you don't want to do now. You don't need to build the liver. You don't need to build the brain. They're already there. So transcription must be somehow regulated. Regulation of transcription is a huge topic, as you can imagine. We do this every day. Every time we have a response to a given stress, or simply, you know, physical activity, non-physical activity, mental activity, we need different proteins. We need something different, so we have to regulate our activity. Generally, what happens, one of the main, classically, now there are so many, but classically, one of the main factors in regulation of transcription is the phosphorylation. So this means to add or remove phosphate groups from a given protein. Generally speaking, phosphorylation, so the addition phosphate groups to a protein activate a given protein, so can make this protein active, for example, go into a translation, and instead the dephosphorylation of a given protein, like in this case, can also be like that. I mean, in this case, you see an inactive protein becomes active because it's dephosphorylated. And finally, you can have in some cases, in some other cases, instead, the quaternary structure of a protein, which has one protein which acts as an inhibitor of the other protein. If you remove the inhibitor, this protein now becomes active. So these are typical classical mechanisms for regulation of transcription. Another mechanism which is now becoming instead extremely important is the so-called methylation. So you see here, this is now hypermethylated areas, and this is also strongly related to these are. If you go from hypermethylation to hypomethylation, and in this case from hypomethylation, so low methyl to high methyl, this is related to the change of expression of the genes into cancer. So you see when you see CpG islands, this means C-phosphate G, cytosine phosphate guanine sites, that are methylated in C to form what is known 5-methylcytosine. Okay, so and TSG, for example, is a tumor suppressor gene. We will see this later, but imagine that this gene is controlling the growth of the cells, suppressing the growth of the cells so that you don't get the tumor. The regulation of these genes can be a hypomethylated region. If you remove this hypomethylated region, you will express these genes and you can have cancer. This is for cancer, but in general, methylation, so this methylation of some forms are essential in the regulation of transcription. And another important regulator of transcription are what are called another type of RNA. We have already seen many RNA. No? We have seen the messenger RNA, the pre-messenger RNA, the transfer RNA, and now we have the microRNA. The microRNA are very small RNA molecules, only 22 nucleotides. So they're really, really small. And they are non-coding, of course, because it's only 22. And they act via base pairing with complementary sequences within mRNA molecules, usually resulting in gene silencing via translational repression or target degradation. The human genome may encode over 1,000 microRNA, which may target about 60% of mammalian cells. So in other words, the genome can code microRNA that are now targeting the RNA and repressing the translation of a gene or enhancing the translation of a gene. This is, you see, a chain of how microRNA can work. So microRNA, if you look at microRNA, you understand what are we doing to regulate the expression of the genes. So let's see again, let's learn a little bit about these words. Now we have the number of genes that makes up what is known the genome. The genome is the set of all human genes at about 30,000. Here in this slide is 25,000, but I think the most likely number is somewhere between 20,000 and 30,000 genes. From the genome, you get the transcriptome. So the transcriptome now is done by the RNA, the set of the RNA that you can do from the genes. And they are more than the genes. Because I told you, I mean, from the gene, from the same gene, you can do different transcripts using, for example, alternative splicing. So you go from some 30,000 genes to some 100,000 transcripts. Now, these 100,000 transcripts are still much less than the total proteome, so the different proteins that we have in the body that are more than 1 million. And this is now, this final step is done by post-translation, what is called post-translational modifications that we have seen, like microRNA, for example. Using this post-translational modification, you can go up to over 1 million different proteins. Okay, in the final slides here, I just want to say a few words about proteins. I told you how important they are. They do almost everything in our body. They are structural proteins like collagen. They are transport proteins like hemoglobin that are so important, you know, that we breathe and we transport oxygen. Hormones like insulin, you know, that hormones is also quite important, among my favorite proteins, especially like testosterone for us and estrogen for women. Receptors, those are essential. These are those membrane molecules that bind to other proteins and tell cells what to do. Motor proteins, these are the proteins that make move the cells, or eventually also muscle. They are the fibers that pull the muscles. And finally, the enzymes. The enzymes are actually probably the most common and most important proteins of all, because no chemical reaction in life would actually happen without enzymes. I mean, would happen, but in million years. The enzymes are the catalyst that make the reaction happen at extremely high rate when we need them. We have seen already many enzymes. The RNA polymerase, which is actually the enzyme that makes the RNA, is an enzyme, of course. The structure of the protein is also quite complicated. You have a primary structure, which in the primary structure is the sequence of the amino acids. You have 20 amino acids, and then you make a primary structure. But then this is a string. Then you can have a secondary structure. Secondary structure, you see, is very often done by the so-called alpha helix, as shown here, or the so-called beta sheets, as shown here. These are called secondary structure. Then you have a tertiary structure. So now you take the alpha helix, the beta sheets, you put them together, and you make a 3D protein, like collagen here. And then you can have a quaternary structure where essentially you put together different tertiary structure of proteins to make a quaternary structure protein like hemoglobin here. So the proteins, as I said, are this is a very nice protein. Here you see a 3D reconstruction of a protein. Look how complicated it is. From you see inside the secondary structure, you see some beta sheets, some alpha helix. This is a simple English. It's a glycolase 1. There are 2 zinc ions that are. This is also why even heavy elements are important in our body. These are trace elements, very rare, but in enzymes are very important. 2 zinc ions that are needed for the enzyme to catalyze its reaction, as shown as purple spheres. And the enzyme inhibitor called S-hexyglutathione is shown as a space-filling model, filling the 2 active sites. So this is, you see, the structure of proteins, as you can see, is tremendously complicated, but this is one of the most important topics, for example, in pharmacology. In pharmacology, you want to study the structure of this protein because eventually you want to inhibit these proteins in case of disease. So they are essential, as I said, for catalyzing reactions in the cell. This was clear since the beginning that there was no life without enzymes. This was very clear. And the first model instead, in fact, is from the 19th century, the so-called lock key model, where essentially you have the enzyme that fits a protein as a lock fits the key. More recently, let's say in the 20th century, the best model used is the so-called induced fit model, where essentially it's not really like a lock key. There is the enzyme can adapt to the structure of a protein. There are infinite examples, but two proteins that I want you to remember because they are so important. We have seen in the regulation of transcription, the phosphorylation and dephosphorylation. This is one of the main mechanisms for the regulation of transcription. The proteins that do this job are called kinases and phosphatases. So the kinases add phosphate groups, and the phosphatases remove phosphate groups. They take the phosphate group from the ATP molecule that, as you know, is the energy molecule of the cell. Why the energy molecule of the cell? Because they actually work. The power plants of the cells are actually the mitochondria. This is where the ATP is synthesized and then can be degraded in ADP, giving energy to the cell. The mitochondria is probably originally an independent cell because it also has its own DNA and ribosomes. So it was really independent. It was then phagocytated somehow in the mammalian cells because of its ability to do aerobic respiration, to generate ATP, and then to give the energy to the cells for the life of the cell. And another protein that is important for the structure is the one for the cytoskeleton. The cells also have a skeleton, and this skeleton, you see microtubuli and microfilaments, are done by this protein, and they are important for many functions: movement, stabilization, transport of organelles, muscle contraction, of course. The chromosome movement and cell division, this is important. We will see mitosis. To move the chromosomes, to divide the DNA in the two cells, you use actually the microfilaments in the cells. The cytoskeleton plays a very important role there. And finally, the cell-to-cell connections. I mean, as I said, all the proteins that are on the surface make the connections to the external world and cell-to-cell. So, for example, the gap junctions that are real junctions that put the cells together are channels that join the different cells in a tissue so that the different cells can communicate. That's about it for now. In the next lecture, we will start to see, we have now seen the different elements of a cell. In the next lecture, we will see how these different elements make up the cells, starting from viruses that are not cells. They are even not really alive by most standards, to the prokaryotes that are bacteria, finally to the eukaryotes that are our cells. Thank you.